Good morning, everybody. It's great to see you, and thanks for joining us the last Wednesday of the month. If it's the last Wednesday of the month, it's our West Coast webinar. So we have the pleasure today of having Dr. Rana Ghosh speak for us. He comes from the University of Texas at Austin. Obviously, Texas has been in the news lately these last few weeks, so we're especially grateful that Rana was able to agree to speak with us today. So as a reminder, my name is Kim Killian. I wanted to introduce the North America West team. Angel Galarza is our regional sales director located in San Francisco, along with Ian Villamagna, who is his field application scientist. As I mentioned, I'm Kim Killian, regional sales manager for the Southwest, located in San Diego, along with my cohort, Viet Nguyen, who is my field application scientist, also located in Southern California. So thank you again for attending. We look forward to you attending future quarterly events. Our next journal club, which is the second Friday of every month, will be on March 12th, and it's at 10 a.m. Pacific time, noon Central time. And then join us next month, March 31st, again, the last Wednesday of every month, for our final Texas series. This quarter we had 3 Texas speakers because Texas is a huge territory for us. So again, thank you for your time and your participation, and I'll hand it over to Dr. Ghosh.
Thank you, Kim, for that introduction. I'm happy to be able to share some of our work with you all today. And here's my contact information below. Before I get started, I'd like to thank the main people who worked on the project, Dr. Jasmine Leal, Dr. Hairi Zhang, and Melis Soto. And with that, so just to give a brief background, my lab is very much interested in understanding how we can overcome the extracellular and cellular barriers to actually improve gene and drug delivery. And today, what I'll talk about is one story where we're looking at cystic fibrosis. So we're looking at how we can achieve pulmonary delivery to overcome, to effectively deliver nucleic acids there. And as you see, I put kind of a laser pointer so you all can be easy to follow the slides. So briefly, cystic fibrosis is a inherited recessive genetic disease with over 2,000 mutations, some which have functional effects and some that don't. And this is mutations of the cystic fibrosis transmembrane conductance regulator gene. And so with the CFTR gene, and they have these mutations. And so this affects, and this is kind of data information, but there's more than 30,000 in the US, and that's increased, and there's more than 70,000 worldwide. That carries. As you see in this kind of cartoon, that the median predicted survival age is only about 41.6 years. Now, briefly, what happens is this is sort of a portion in this cartoon. You have a portion of the CFTR gene. And there are, like I mentioned before, there are different mutations that occur, and this could be mutations, deletions, insertions, and they lead to various downstream effects. So as we see, you can have things where in the most prevalent mutation, which is the deletion of 3 bases, which leads to deletion of the 508, you get a immature dysfunctional protein, which then leads to the protein degradation. And then you can have other things where you can actually have no protein, due to missplicing, you get your RNA out of frame. You can have unstable RNA leading to no protein being produced. And why is this important? Because with these mutations, what occurs is on the in terms of the disease phenotype, it's a very systemic disease actually. And so on the left, you can see the various kind of organs. So pretty much with the mutation, you have a defect in or lack of defect of that CFTR protein. This protein is a transmembrane protein that's present on the apical side, of your epithelia, which is lying sort of mucosal tissues. So this systemic disease, you have, things like your skin. What happens? Your skins will produce salty sweat. You actually have blocked pancreas ducts, and then even your intestines, because of this defective protein, you have, in general, what happens is you get impaired ion transport, chloride transport, and fluid transport. And then the byproduct is you have sticky mucus surrounding these tissues. And so, in the intestines, you will be able to absorb your nutrients. And probably most prevalent, what we think of is the airway epithelia. So if we see on the upper right, you have normal airway. You have, you'll typically have a thin layer of mucus, and you have mucociliary clearance, essentially a cilia lining the epithelium that will clear away the mucus. You have constant turnover. Those with cystic fibrosis, there's a thickening of the mucus as shown on the bottom with green, the thickening, and you actually then can get bacteria that can remain because you don't have essentially the mucus is thick, it's dehydrated, and impacts the cilia and prevents it being cleared. And then bacteria can accumulate, and you can even have blood, and this actually leads to impaired breathing. And so a lot of times we think of this kind of airway disease, but it is a systemic disease. So what are sort of the various strategies? So there are small molecule approaches to essentially restore the function of the CFTR protein to get more, essentially restore the chloride transport and kind of get rehydration of the mucus and then get clearance. So that's more of a functional. To get at more kind of cures can you correct it on the genetic level. So there's things where you can add oligonucleotides to correct things like splice defect. And so you can then produce a functional protein. One is also gene editing, where you can actually kind of use those molecular scissors like CRISPR and Talons, zinc fingers, to actually make the edits to correct those mutations to get a functional, mRNA, and thus get your functional protein. Also, what you can do is you can do gene replacement, where you can deliver DNA or actually deliver mRNA to restore the functional CFTR correct protein. It's kind of correct sequence and then get the correct protein. So those are various strategies. Now, mRNA, as we're aware of kind of with the various COVID vaccines, has been quite promising. So this is sort of the cartoon of just native mRNA with a 5' cap on the 5' translated region, the coding sequence for your gene of interest, stop codon, a 3' and untranslated region, and a polyadenylated tail. And so this is sort of the native structure. What they've shown is you can actually make in vitro versions of these, synthetic versions, where you can reduce the immunogenicity and improve its half-life. And it's very advantageous because if you can deliver, so if we, in kind of the cartoon, if you can deliver the mRNA, it's advantageous because it doesn't integrate into your genome. And then as long as it can be released from the endosome into the cytoplasm, it does not need to get a nucleus. So you have the mRNA, which can then be taken by your ribosome to actually then get translation into your functional protein. So mRNA offers these advantages of not having to get into the nucleus. It's transient, so you can actually do repeated dosing and not integrating into your genome. And so it's been exploited, as we're aware of, for very therapeutic purposes, such as the COVID-19 vaccine. So there are challenges, though, to get the mRNA. So if we want to deliver CFTR mRNA, or maybe do CRISPR-Cas9 kind of CRISPR mRNA, kind of gene editing mRNA delivery, there are challenges. And so in this cartoon here, one of the things is there's those extracellular barriers. So how you could actually get the mRNA degraded, or you could actually get immune response, even though you can actually make modifications to mRNA to prevent this, and you can get degradations. So as a result, there's been efforts to try, can you encapsulate and protect that mRNA for a more effective delivery. Now, there are these, however, if you want to deliver it, the challenges with cystic fibrosis, you have these extracellular barriers. So one of the things I mentioned is you have thick mucus. As we see here in the EM on the left in normal, on the right, you have healthy mucus. As the laser pointer show, you have this pore size, you have these nice pores, you're gonna get clearance by the cilia, as shown on the left here. Now, with patients with with cystic fibrosis, you see this thickened mucus as seen here on the left-hand side. And so you have a decrease in the pore size, and also in the bottom cartoon, what you see is the mucus with people with cystic fibrosis is more viscoelastic. There's change in pH. There's changes in kind of in the ionic strength, and there's changes, and it's heavily charged because of the mucus is very kind of negatively charged, and because you have this barrier which prevents really a lot of drugs, almost up to 99% of small molecule drugs are unable to penetrate through this barrier. And so if you now want to develop delivery systems, how do you do that? Well, there's been efforts by others and ourselves to try and use polyethylene glycol, or PEG, to densely coat the surface of drug gene carriers to then deliver across. So what happens, what I want to show here is the PEG on its surface essentially it's a muco-inert coating. It allows for diffusion through the kind of the mucus barrier. And on the upper left, what we have is if you have a particle that doesn't have a PEG coating and you deliver it to the airways, you actually get poor distribution kind of in the bronchioles, in the airway space, and you get aggregation. Now, if you coat it with PEG, these carriers, you'll get more uniform distribution, as we see on the fluorescence micrograph on the bottom. You get more uniform distribution. And then on the right, what this is effectively showing is on the bottom right, as you if you coat your drug gene carriers with PEG, you get better diffusion through the hydrated pores of your CF barrier, your mucus barrier. Whereas with uncoated, up on the upper left, uncoated particles, what we see is the diffusivity can be 1,000-fold less. And so this is the advantage of using this PEG coating. And we have shown this also. So don't want to belabor the point. Now, if we come back, we have that extracellular barrier, the mucus, but then the challenge is, well, how are we going to deliver that mRNA? And we talk about that using kind of carriers to encapsulate that mRNA to get it through the extracellular barrier and then intracellular to the into the cell, so then it can get into the endosome and then have effective endosomal escape and release into the cytosol for then protein production. So there have been different carriers that have been used for cystic fibrosis. A lot of early efforts have been focusing on viral vectors on the upper left, such as adenovirus and AAV. While they have their advantages, viruses are naturally evolved to infect cells, especially even our airway epithelia. However, there are challenges with, say, immunogenicity. And in the case with these viral vectors, you can have packaging constraints. So with AAV, it can only package about 4.7 kb, so it can limit how much of the CFTR gene can be packaged. But they are still very promising. But there are efforts to kind of look at non-viral vectors. They offer the advantage of reduced immunogenicity. You don't have the issue of packaging constraints, and you can deliver various types of cargoes of different sizes, such as plasmid DNA, your mRNA, or even, like I said, kind of your CRISPR-Cas, your gene editing components. So you don't have those size constraints. And so we're very much interested in the lipid nanoparticles, as many of us are. And lipid nanoparticles, there's been extensive document literature about its promise to encapsulate nucleic acids, as with kind of the various COVID-19 vaccines. and various actually in the CF field, there's been work trying to use kind of lipoplexes and liposomes to try and deliver, and they've entered clinical trials. However, there are still challenges to improve their really for airway delivery to improve the functional output, and so that has been a challenge. But there's been ongoing work. So there's of course with lipid nanoparticles, they're kind of non-toxic, and they're very well characterized. So very much interested. However, one of the challenges with using non-viral systems, well, any carrier system, is the effect of aerosolization. As I mentioned before, while CF is a systemic disease, most efforts are trying to improve, kind of restore the airway function so you can breathe. And so one of the challenges with using these carriers is eventually you'll have to put it into, you'll either want to put it in nebulized form, so you want to aerosolize it, or put it in a dry powder form where then you can then inhale it. And then once it inhales, it still has to penetrate all the way through, get to the bottom parts of your kind of your airways where they can penetrate, as we see here, the mucus, excuse me, go through cilia and get into the affected airway epithelia that has these affected mutations. Now, one of the challenges with aerosolization, you have the shear stresses that, due to aerosolization, that your carrier is subject to, and even your nucleic acid. So that's something we need to think about before we, to consider. So this gets at our ultimate sort of question is, there's so many barriers. The aerosolization aspect, getting through the mucus barrier, and then, excuse me, the intracellular barrier. Can we develop a way to kind of do a high throughput screen where we can screen different lipid nanoparticles that encapsulate mRNA that are still stable enough to then achieve effective mRNA delivery in cell lines, but also in in vivo, in animals? So our general strategy was to use a design of experiments approach. And the idea here is we would develop a library of different formulations, of different lipid nanoparticle formulations. And so in the cartoon, as I'm circling, in our lipid nanoparticles, we're using sort of kind of the very canonical components. So we have our cationic ionizable lipid that we use. We have our PEG-lipid to provide, in this case, for pulmonary delivery, to get that mucus penetration. We have our helper lipid, our helper phospholipid, and excuse me, we also have our one moment. We have our structural lipid, our cholesterol. And so we have these four components along with our mRNA. And the idea is, can we combine them at, make different formulations of different amounts, ratio, and then look at how effective they are. So we want to make this library. The reason why we want to use a DOE, a design of experiments approach, is we, wanted to really look at all of our parameters as a whole, as opposed to just kind of changing one factor at a time, as opposed to changing, say, modulate just the our ionizable cationic lipid. Can we look at all the parameters at once? And after that, we would characterize their properties. After formulating it, we would characterize their properties, see how nebulization or aerosolization affects those properties, test it on cell lines, and then see if we have any lead candidates, see how they'll work in vivo. So we came up with, based on the literature, with lipid nanoparticles delivering siRNA and mRNA, we came up with our components, and we came up with sort of a lower limit and an upper limit. And so these are sort of all the components and kind of the range that we're looking at in terms of our molar ratio. So the contribution of each component to the formulation. And so this was pretty much these values, these lower and upper limits were taken from the existing literature of what's been out there, what's been published. And so on the bottom is sort of the various compositions of different phospholipids we tried, or helper lipid, the different PEG-lipids that we tried, and then those compositions. And so we kept, we just used basic cholesterol, and we actually use the MC3 ionizable lipid that's been very heavily used out there. So, for those who aren't aware, we actually use the Precision Nanosystems nanoassembler to achieve high throughput rapid assembly of these formulations. So we made 18 formulations, and this allowed for very rapid mixing where we could take our lipid components in the ethanol phase, our organic solvent, and then our mRNA and our aqueous solvent with the rapid mixing make our particles. And so this allows for kind of rapid assembly of our formulations, which allows for very easy testing downstream. And since we're using the assembler, this allowed us to actually achieve quantities that were sufficient for not only cell testing, but also animal work, as I'll show later. Okay, so before we did the DOE, what we kind of tested the different formulations, we wanted to figure out first, what is the fix? We wanted to fix our N to P ratio. This is essentially the amount of amines to phosphates, and really what's affecting the number of really our particles, our lipid nanoparticles, to the ratio of phosphates due to the mRNA. So that N to P ratio. We want to figure out what is the optimal ratio. We want to keep that number fixed before we start changing all the other components in our DOE. So what we did is we actually using Alnylam's Onpattro formulation. We used all their components and we just changed the ratio. Instead of sRNA, we used mRNA and we changed the molar ratio of our N to P ratio of our lipid nanoparticles. So we went from 6:1 to 200:1 ratio. And what we did is we made different formulations, mimicking the composition of the Onpattro formulation. And we delivered a GFP reporter, mRNA reporter, and we looked at cell, essentially expression of GFP through flow cytometry. And what really the take-home message here in this graph is at 15:1, we saw highest expression of that reporter and also the greatest intensity. So we use that 15:1 ratio going forward in our subsequent DOE kind of experiments. So this is just to refresh. This is our kind of the different formulations we did at that N to P ratio of 15. So after formulation, we want to characterize how stable are these lipid nanoparticles, and we want to characterize their kind of physical chemical properties. So on the upper left, in blue, is our formulation after day one, just in storage at 4 degrees. We just measured, what is the size. And typically, for all our formulations, we get a range of, a little bit under 50 nanometers to about, 120, 130 nanometers. And at day 14, when we kept it in the refrigerator, we see that they're still stable, as seen in red. There's no changes. On the upper right, we see with our zeta potential, when we look at sort of a proxy for surface charge, there's no really any changes from day 1 to day 14. What is interesting is with different formulations, you do see a change in that zeta potential from, say, negative to even slightly positive. So, that is interesting to know. Our formulations were very good at encapsulating the mRNA, where we actually measured the encapsulation efficiency. All except for one had near 100%, and this was before nebulization. Now, when we look at the pKa of our nanoparticle, we see that it ranged from 5.5 to 6.4. This is, comparable, say, the Onpattro formulation had a pKa of roughly 6.44. And this is important if we want to achieve eventually that endosomal escape, so we can have a release of the mRNA for a subsequent, translation. Next thing is, we realize that they're stable. Their physical chemicals are stable. These pre-nebulized formulations. What happens when we take these formulations stored for different days, and how are they in terms of their transfection? How are they able to deliver the mRNA? So we had our mRNA with a GFP reporter, and we dose in different wells 100 nanograms of our GFP in our formulations. And after 24 hours, we looked at transfection by flow cytometry of formulations that were stored for different amounts of time. And in general, the take-home message is, they were the formulations were able, those that are able to transfect well, they were able to maintain that transfection in this sort of heat map. But there are select formulations that are better at transfecting, and I'll go into specifics on certain ones in a little bit more detail. So not all formulations perform very well. As we see, the ones in red are ones that have kind of low levels of transfection, whereas the ones in green had higher levels of transfection. Now, the one thing I mentioned, we did this all kind of before nebulization. That's all nice and good. What happens when you nebulize it? So we use this aerogen nebulizer to create essentially effectively aerosols. We collected these aerosol versions of our formulations, and then we collected them, and then we went back and ascertained how are the physical properties changed. So one thing to note is with, after nebulization, when we look at by dynamic light scattering, we can see that all of the sizes increased of all the nebulized particles, those formulations after we nebulized them. So there's a change there. One thing that is also interesting to note, though, is our zeta did not change. So the surface charge conceptually was stable. But then our encapsulated efficiency, you do see, before nebulization in blue, you see that's very high, near 100%. But then after nebulization, you see that decrease, which suggests that, also with the change in size, that you get increase in size, but you're also getting a breakup of these particles, and potentially the mRNA may be even degraded. So nebulization does affect those physical chemical properties. But once we take these properties, are there certain formulations that are still effective? So we know aerosolization can have an adverse effect, but are there any still good candidates after formulation that can still be effective in terms of functional, transfection? So what we did is we had our pre-nebulized versions of our formulations and our nebulized formulations, and we did incubate them in two different cell types. We had it just on human embryonic kidney 293 cells, and we also had it on Newly cells, which are kind of lung epithelial bronchial cells that actually possess a 508 deletion of the CFTR. So this is kind of the most prevalent mutation present in CF. This cell line possesses it. And so what we did is we incubate our nebulized forms and measured expression of the reporter by flow cytometry. And so, in general, after nebulization, as we see in the 293s and hex, there is decrease. You see blue is the pre-nebulized or unnebulized lipid nanoparticles. After nebulization, you see a decrease in general for all of them, and with both cell types. So there is loss. However, one thing to note there are 4 candidates. So F2, F8, F11, and F17. While there was slight decrease, they had the most minimal loss of transfection. And we want to then proceed with these formulations going forward. So while aerosolization impacted its size, their size, even its encapsulation efficiency, it was still was able to achieve better transfection than these other formulations. So what we did next is, it shows some promise in some cell lines. How is it in kind of healthy mice, which actually have, mucociliary clearance, so they're actually able to clear away. So we're not testing on mice that have cystic fibrosis, but ones that actually have clearance. So we actually need something that can penetrate. We deliver through the pulmonary route, but then it needs to be able to penetrate and get into the lungs before it gets cleared away. So this is actually, in some ways, a bit of a harder challenge. So what we did is we took our different formulations, our lead candidates in pre-nebulized and nebulized form, and we took our aerosolized form, we delivered intratracheally, and they were delivering a luciferase reporter. So then we can measure with kind of luciferase imaging. So we administer it intratrachely. 6 hours after administration, we sacrifice the mice, we harvest it, and then we did luciferase imaging and measure the lungs of the animals that were administered with these formulations, and also other tissues. And so what I want to show is kind of first in this not cartoon, in the images on the right. In our pre-nebulized form, we see, signal and the signal is indicative of the luciferase protein, the reporter, and kind of the light being expressed as a byproduct of luciferase imaging, or imaging for the luciferase reporter, excuse me. And so we measure here, this signal is indicative that we are seeing, expression of our luciferase. So there's not only delivery, there's penetration of the mucus and getting into cells of the airway and getting expression. And we can see it with all these formulations. What is interesting to note is after nebulization that we still are able to see signal persist. And if we go here, if we actually try and quantify it on the left here, with our formulations, there's actually no significant difference between our pre-nebulized forms. When we actually try and quantify the signal, there's actually no difference between our pre-nebulized formulations shown in black and our nebulized form shown in the pattern bar graphs. So there's no difference between our different formulations. So we're actually getting a signal. And then compared to our when we just deliver naked mRNA, there's no kind of detectable signal when we deliver it through the kind of pulmonary route. So what is interesting is, this gets into the issue of, we saw before and after nebulization, there were size changes, there's losses in encapsulation efficiency, but it was still sufficient to achieve expression. And even with those losses due to nebulization, that you did not see, it was still effective in terms of reaching the cells of the airway to have some functional expression. Also, one advantage, which did not mention before, is by doing pulmonary delivery, you get this localized delivery where you can, avoid potential off-target uptake in other tissues. So as we see on our images on the right, when we look at the heart, the liver, and the kidneys, we do not see, at least by luciferase activity, we do not see any signal present in these other types. As we're aware, a lot of times with systemic delivery of nanoparticle formulations in particular, a lot of lipid nanoparticles, you will get, significant liver accumulation. So this also raises the advantage of doing kind of pulmonary delivery. So, we have this information, but what parameters could be involved? So what I want to highlight is we actually went back and did sort of a jump analysis to look at, we did this DOE, we had all these formulations. We could go back and now tease out parameters and how they may affect physical chemical properties and also affect kind of transfection efficiency and kind of delivery of our reporters. So one thing to note is, excuse me, is of course, before nebulization, what we see is, as we increase the PEG-lipid molar ratio, so in your formulation, as we increase the amount of PEG-lipid that we're including, the size goes down. But what's interesting to note is initially, before nebulization, shown in B, there's really no difference of the type of PEG-lipid. So regardless of what PEG-lipid you use, DMPE-PEG, DMPG-PEG, it had no effect on the physical property, on the size, before nebulization. After those, we see in C, after you nebulize the particle, we see the DSPE-PEG gets much larger. If we look at kind of B to C, it goes from 60 nanometers all the way to over 1,000 nanometers. Sort of the median is the size of these particles, and that's important because to to penetrate the mucus barrier to eventually get into the affected cells in CF, typically what's been found in the literature is that mucus barrier, there's roughly a mesh size where particles of 300 nanometers or less, especially if you're muco-inert particles, such as PEG-lipid particles, they're able to penetrate through. But anything larger, you can actually get impaired diffusion. You will get that penetration. So this is where nebulization impacts those physical chemical properties, which can have an potentially adverse effect. And as we notice, if we come back to our sort of DOE, our top 4 formulations that we eventually did our animal study with, none of them, as we highlight here, none of them had DSPE PEG. So none of them had DSP PEG as shown here, which says that that could play a role. The choice of PEG-lipid can play a role because before nebulization it behaved one way. After nebulization, the properties change. And you have to be mindful of that. Now, before and after nebulization, there are changes. So we think that PEG-lipid, though, the amount of PEG-lipids, regardless of before or after nebulization, still plays a role. So what we have here is our various PEG-lipid. As we on the x-axis, as we increase the amount of PEG-lipid in our formulation, regardless of the formulation, there's a decrease in expression. So you can think about it as you're creating more PEG on the surface, it's unable to be kind of taken up by the cells. And this has been shown with a lot of people studying PEGylated nanoparticles and cell uptake. So this is well known. This is still maintained after nebulization. As you increase the PEG, you still have that decreased amount. But we have to be careful. There's a balance because we still need enough PEG to have that mucus penetration. And what is interesting to note is our best performing particles, those top 4 formulations, they had about 1% PEG. So they're up here. So you still need some PEG, but maybe you don't want too much because you won't be able to get that eventual, not only penetration, but cell uptake that you need to then release the cargo. But before and after nebulization, we think PEG-lipid still plays a role. So that's a parameter that we have to maybe keep in mind. And because it seems to correlate. Now, we don't necessarily know if it's causative, but it correlates with changes in transfection. Also, another thing we've noticed in our analysis is before and after nebulization, that there are, the type of phospholipid that you use, that helper lipid can play a role. So before, you would think, before nebulization, if we use DSPC compared to the other lipids, you have decreasing GFP expression. So we think before nebulization, maybe it plays a role. Maybe not, because after nebulization, there's really no statistical significant difference between the different types of phospholipid used. So it may not play as much of a role, but this is more correlative. But we see there are changes. This is just to emphasize that really before and after nebulization, there are the changes due to nebulization in kind of the components used of PEG-lipid or phospholipid can affect that ultimately your delivery of your nucleic acid reporter. So those are just some of the highlights from our analysis of what we saw. But what I wanted to conclude was, using DOE allows us to kind of look at all the variables. So in our lipid nanoparticle formulation and with others, you have all those components, right, involved. You can introduce all those variables and still kind of look at what could be, what is meaningful in terms of transfection and delivery. The nanoassembler allows us to kind of rapidly assemble and make these formulations, which is very advantageous. And then using our DOE approach, we're able to kind of, and subsequent characterization and kind of assays, we were able to find aerosolized formulations that could transfect in cell lines, but also in healthy mice. However, really, this is really an initial step. there are really more questions that actually come from it, which we're very interested in. So one thing is, how does, we know that aerosolization impact the physical properties and subsequent function, but how? Why does aerosolization, maybe the sheer stress due to the aerosolization of the particles, why does it affect some formulations and not others? Also, what we don't know is with the aerosolization, how does it degrade the mRNA? Does it likely has an effect, even though we're encapsulating it with a lipid nanoparticle, how does it affect the mRNA stability and subsequent transfection? We don't know that yet. Also, that analysis I showed was really looking at, a single parameter and how it changes before and after nebulization, such as type of phospholipid or PEG-lipid, um, um, and how does that affect, or how, before and after nebulization, how does it correlate with transfection efficiency? What really the DOE, what we haven't really taken full advantage of, what we're doing now is looking at how do these multiple interactions, you have these different components between, say, cholesterol and the PEG lipid, or PEG lipid with the type of phospholipid. What are the parameters that are truly driving the transfection? I mean, what also affects formulation stability, but what really affects transfection? Maybe some things, as I showed before, the type of phospholipid post nebulization, it did not seem to play a role. It did not matter what phospholipid you used, but maybe there's some interaction that with some other parameter that is actually critical to driving that transfection efficiency. One of the things that we have to keep in mind is we have to balance. Really, we need our formulation to be stable enough when it's aerosolized, but then it has to then be able to go into our lower airways, penetrate the mucus, but then be flexible enough to disassemble, once it gets taken by cells, to disassemble, to have that early endosomal escape of the mRNA for to get your functional protein made. So you have this balancing. So we are really trying to figure out what are the true parameters that are driving our ultimate goal of functional protein delivery, or protein expression, sorry. And then a few things that are also important is, since we're doing pulmonary delivery, what is the role of the bronchoalveolar fluid? Essentially, it can form a corona on your formulations, and does that redirect its fate in terms of going through the mucus and getting the cells? Similar to systemic delivery, kind of the protein corona you have to worry about there. And then finally, we want to look at more disease-relevant models. As we did test in sort of cell lines and in healthy mice. In the case of cystic fibrosis, what we'd like to do is you can actually develop in vitro models where you can do air-liquid interface of cells or even organoids, which better recapitulates kind of the air-liquid interface that you have when pulmonary delivery. And also there are testing in more disease-relevant models in animals, such as there are kind of mice, pig, rat models, ferret models that better recapitulate not only the genetic aspect of cystic fibrosis, but also the phenotypic aspect, where the byproduct of getting that thickened mucus. And so these are things that we would want to test with our formulations. But like I said, I think there's a lot of questions to understand in terms of what parameters are really allowing for good enough stable formulations after aerosolization, and then good enough to then actually get proper mRNA delivery and subsequent expression. And with that, I'd like to thank, like I said, Jasmine here, and Melly, who worked with our collaborator, Hairi Zhang, and Dr. Smy's lab to carry out this work and our funding sources. And with that, I will be happy to take any questions.
Thank you very much. And I think we're really getting close to the top of the hour. So I'd like to let our attendees also have some time before their next virtual meetings.
Sure, yeah.
So really, we'd like to thank you very much, Rana, for the wonderful presentation, and really for joining us today, especially with how Texas is right now.
Yeah,
Kim, would you like to close us out?
Oh, sure. Rana, were you going to say something?
Oh, yeah, I was gonna say if there were additional questions, I was just gonna put up, sorry. If people had other questions, they could just my email is listed there. So
Okay,
thank
great.
so
We
you
have
can
had
if
some
people can
requests for your slides too, so please feel free to copy down Rana's contact information and reach out to him. We do have a YouTube channel up now, which is exciting. So you can find this webinar as well as our other webinars on YouTube, and we have recorded this as well. So there's lots of options. And again, thank you, Dr. Ghosh. It was a great, stimulating conversation. We had tons of questions. That's always a great sign for participation. Please join us again as a reminder. Join us in March for our Journal Club, the second Friday of every month, starting at 10 a.m. Pacific, noon Central. And then Christian Buddha from Texas A&M will actually be hosting our next webinar at the end of the month, the last Wednesday of March. So please join us and happy Wednesday. Have a great week and take care, everyone. Stay safe.
Thank you. Take care.
Thank you.